A very good morning, class. Today we are going to start the second disaccharide, and that is maltose. Its general formula is also same as sucrose, that is C12H22O11. But what is the basic difference between the between the sucrose and the maltose? That is the glycosidic linkage that we will do. Okay, before that, I just want to tell you that it is a reducing sugar. Although it is a disaccharide, that means the CHO must be bonded, but still it has the capability to reduce the tolerance reagent and Fehling solution. That's why it is called reducing sugar. So this can be it is never been asked, but if it is asked, so you can write that although it is a disaccharide, but the CHO group this is capable to reduce the tolerance reagent and the Fehling solution, and that's the reason it is known as reducing sugar. Okay. Second thing, if you check, this is we have done on the very first day of this chapter, that maltose on hydrolysis gives 2 moles of glucose. We have done 3 disaccharides. One was that sucrose, which on hydrolysis was giving glucose and fructose. Next was maltose, that is giving 2 moles of glucose, and third was lactose, which were giving 1 mole of glucose and 1 mole of galactose, isn't it? Now, if we talk about that how to write the reaction, we can write in this way. There is no issue. We can write in 2 moles. So both are glucose, so that's why the same general formula, same functional group. So we can write 2 moles of glucose, that also. Where is the glycosidic link is present in this maltose? It is present at C1 of 1 alpha glucose and C4 of other alpha glucose. So to be very precise, we can say that in this glucose, it is not anomers. They are not anomers. They are exactly same. That OH group is on the same side, and that is inside the ring, isn't it? Both are alpha. But what is the basic difference? That it is C1 C4 linkage. So sometime you can see that it is 1,4 glycosidic linkage also said as. So you should understand that C1 or C4 or 1,4 glycosidic linkage, what does it mean? It says that 1 alpha glucose and other alpha glucose will combine together by C1 and C4 carbon. Okay, let's see this how to work. See this one, if we check, this is the 1 glucose molecule, and since glucose structure is resembling with pyrano, so we can say alpha D glucopyranose, or this is pyranosyl bhi keh sakte hain hum ise. Okay, so alpha, that means we know that OH should be inside, that means नीचे की तरफ होगा OH, isn't it? And this is D, which resembles with the glyceraldehyde structure, and pyrene, because it is the Haworth structure is resembling with the pyranose, so that's why it is called pyranose. Okay, and this one is the another glucose molecule. Here it is 1. This is called 1. This is 2, 3, and 4th. So this is also alpha, because we can alpha. How can we check? On the first carbon we have to check. If both are on the same side, that means it is alpha. That means inside the ring, or in the Haworth structure downward. Okay, so this C1 and C4, here it is supposed to be OH was there, yes, and here also. So what will happen? This like this, yes, and so this H and this OH will come out as a water, and this oxygen. Now this oxygen will act as a linkage between this. Okay, so this oxygen left out, which is now acting as a linkage. So this is also called 1,4 glycosidic linkage or 1,4 linkage. So what you have to learn in this, it is a reducing sugar. That is the 1 point. Another point, on hydrolysis it gives 2 moles of, and the third point is C1 C4 linkage. But this is the key point. You can write it over there as a key point. This is the detailed structure, how it forms anomeric carbon. Sometime it can be asked that in glucose, which one is the anomeric carbon? Anomeric carbon, that means the carbon at which OH position is different in its 2 different structure, in its 2 different rotation. So what we will say, that is carbon 1. Carbon 1 is the anomeric carbon, because if OH is inside the ring, it will be called alpha, and if OH is outside the ring, it will be called beta. So just on first carbon we decide. So that's why anomeric carbon is carbon 1. Okay, so this is alpha D glucose, glucose always in that dextro. Here also, this is 1,4. What will happen? This is the anomeric carbon, and this is like alcohol in presence of H. It will come out H2O, and this oxygen will be left over there. So this will act as a linkage. So 1 and 4 carbon it is going to combine, linked. So that's why it is called 1,4 glycosidic linkage, and this is the structure of maltose, which is a disaccharide. This OH group, as I told you, that if it is downside, that means it is inside the ring, and that means it is alpha. So this orientation of this OH group identifies this disaccharide as alpha maltose. Alpha. Why? Because OH is downside. That means it will be alpha man. अगर यही OH ऊपर की side होता तो what it will be? It would be beta maltose. Got it? Okay. So now the next thing, if you see that this is the third disaccharide, and that is lactose. The same general formula, but the only difference will be the linkage. Okay, glycosidic linkage will be different. So in this case, it is also reducing sugar. So see, out of these 3 disaccharides which we have to study, both like sucrose is only sucrose is a non-reducing sugar. Rest maltose and lactose both are reducing sugar. Although it will never be asked, and it has not been asked, अभी तक तो नहीं पूछा गया है, but sucrose के लिए always it was asked that why it is called reducing sugar. Next, when beta D galactose, I just want to tell you before this, on hydrolysis of lactose, it gives galactose and glucose. We have done, yes. So now I'm just going to tell you what is the difference between galactose and glucose. Galactose and glucose are not anomers. Even like we can say they are epimers. They are not anomers. Anomers carbon 1 पे अगर OH की position different हो, then they are called anomers. But if carbon 4 पे अगर OH की position different होती है, then they are called epimers. Okay, so they are epimers. In this, ग्लैक्टोज और ग्लूकोज में क्या डिफरेंस होता है? वी नो दैट कि सारे OH जो होते हैं ग्लूकोज में इनसाइड द रिंग होते हैं, एक्सेप्ट द थर्ड वन. यस, सो व्हाट विल हैपन, ग्लैक्टोज का स्ट्रक्चर कैसे डिफरेंट होगा? 4th कार्बन OH विल ऑल्सो बी आउटसाइड अलॉन्ग विद द थर्ड तो होना ही होना है दोनों ही केस में, ठीक है. बट 4th पे क्या होता है? ग्लूकोज, इन द ग्लूकोज द OH group is inside the ring, but for the galactose OH group will be outside the ring. That is the only difference. Got it? Okay, so when beta D galactose and beta D glucose combines by C1 of galactose and C4 of glucose, okay, so then it forms lactose. And here I have written that what is the difference on this galactose. In galactose, OH on C4 is outside. Okay, so got it? This is galactose and glucose, galactose C1 and glucose C4. Okay, so here I have, this is the structure of beta D galactopyranose. You can check this carbon 1, 2, 3, 4. 4 पे carbon is outside. Then if it is outside, then only it is upside, isn't it? In a Haworth structure, if in that ring wall structure, if OH group is outside, then in Haworth structure that group will be on the upside. Yes. So since it is upside, we can say that it is a galactose. But in this glucose, you can see that 4th carbon पे OH क्या है? नीचे ही है. Yes. So this OH and this H, here OH is there, no? Here also OH is there. Yes. Before the linkage I'm talking about. And here is the OH. Yes. So what will happen? This H and this OH will come out to form water, and this oxygen act as a linkage between these two. So this is also called cross-linked glycosidic. So lactose is known for its cross-linked oxide linkage. Cross-linked what we say, glycosidic linkage. You can see that it is, one is from this side. Sometime in the structure it can also be shown like this. ये ऐसे भी दिखाया जाता है. And I hope you got it what I am going to say. This is the carbon 4 and this is the carbon 1 of galactose, and this is carbon 4 of glucose. We can draw the like structure is like this. Yes, O This is the O over here, and this is the structure as it is. It will be no change in this. Yes, there will no change in this. सारे जो जहाँ है वही रहेंगे. But in this case also it is like this. Got it? So this is the structure, like cross-linked. And this is drawn in some curve-like structure. This is also called cross. So you have to learn this point that cross-linked glycosidic link is. Cross-linked. Yes. Glycosidic linkage is present in this lactose. So hope it is clear. And on hydrolysis, lactose will give you glucose and galactose. Okay. So this was all about the disaccharide. And this is the structure, like this is beta D galactose and this is beta D glucose. This OH and this H will come out, and here it will form a cross-linked structure like this. The rest is same. Okay. So now we are going to start with the polysaccharides. And polysaccharides, we don't have to go in details, but yes, we have to know, we are supposed to know few polysaccharides. When the large number of monosaccharides join together by glycosidic linkage, they are called polysaccharides, and they act as a food storage or structural material in plants as well as in human beings also. Like starch, you have heard. Starch is the polysaccharide present in the plant, while the glycosin is polysaccharide present in the animals. Yes. So this is the thing, polysaccharides. We have to know which gives more than 10 monosaccharides on hydrolysis. Okay. And there will be glycosidic linkage, because glycosidic linkage is the oxide linkage between two monosaccharides. So obviously अगर 10 होंगे तो उनके बीच में भी तो linkage होंगे ही. So that's why this is called. Okay. So now we are going to study that the first polysaccharide, that is starch. So we all know about like many things about, we have heard this name also. Starch, it is a polymer of alpha glucose. All alpha glucose are there. Yes, it has two component, amylose and amylopectin. This is again going to be an important set of question. First question will be asked, what is the difference between amylose and amylopectin? Okay. Second thing in MCQ can be asked that which portion is the major content of starch? Amylose, amylopectin, both, none of the above, like this. Okay. Third, out of amylose and amylopectin, which one is water soluble? So this three type of question can be asked from this topic only. So let me explain one by one. Amylose is soluble. Sorry, I forgot to write this S. Amylose is soluble in water. It is a long unbranched chain. Since it is unbranched, that's why it is a soluble. Got it here? Okay. So question can be asked that why amylose is soluble in water? Because it is unbranched chain. With 200 to 1000 alpha D glucose, and there will be C1C4 glycosidic linkage only. So amylose, one point you have to learn that it is soluble. Why it is soluble? Because it has unbranched chain. Okay. Amylopectin is insoluble in water. Why? Because it has a branched chain. Why amylopectin is insoluble? Because it is a branch chain. It is a polymer of like polymer hai, but it is a branch chain. Okay. And here also C1C4 linkage will be there in the straight chain, but in branching C1C6 linkage will be there. That means एक कार्बन का C1 और दूसरे कार्बन का दूसरे मोनोसैकेराइड का C6 कंबाइन करेगा. Got it? Okay. Next difference is, next third question I पूछा कि out of amylose and amylopectin, which is the major content of starch or major portion of starch? तो that is amylopectin. ये पूरे स्टार्च का 80 to 85% जो होता है वो पूरे स्टार्च का amylopectin होता है. If I'm talking about 100% starch is there, so out of that 80 to 85% जो होगा that is amylopectin, and rest 15 to 20% is 80 to 85% it is amylopectin. Got it? Amylopectin. and only 15 to 20% is amylose. Got it? This is important point, and I told you that it is three type of question can be asked. One question will be 2-marker, like what is the difference between amylose and amylopectin. You are supposed to write that it is soluble in water, it is insoluble in water, it has long unbranched chain, it has branch chain. You can say that it is 15 to 20% of the whole starch, and it is 80 to 85% of the whole starch, like this. Okay. Glycosidic linkage is present. C1C4 glycosidic linkage is only present here in the straight chain, C1C4 glycosidic linkage, but in the branch, C1C6 linkage. So this is of 2-marker question. Write the differences. MCQ question can be asked, why amylose is soluble in water? Because it has unbranched. Or why amylopectin is insoluble in water? Because it is branch chain. So one question. Second question, out of amylose and amylopectin, which one is having major content of the starch? That is amylopectin, which is 80 to 85%. Got it? Okay. So see, this is the structure amylopectin. So branch chains are there. It's a long branch chain, and here it is unbranched. It's a coiled structure, but unbranched, isn't it? And here it is 1,4-glycosidic linkage. ये straight chain में जाएगा. But if you check in the branch, this is CH2H here, and this is going to, so C1 and C6 bond linkage will be there. So in the branch, but here only 1,4 linkage is present. Got it? Yes. So next polysaccharide is cellulose, and it is the we know bio students and everyone knows about that the cellulose is only present in the plants, and as a cell wall भी present होता है. It's a building material as well as it provides the strength to the cell, isn't it? So it occurs in plant only, and it has a straight chain and consists of beta D glucose. Not so important. It is like not asked, never asked. Okay. So next we can see that this is C1C4 linkage is there. So as like we have done that sucrose and all. So it's same as. Okay. Now the glycosin. Glycosin is the polysaccharide which is present in human beings. Glycosin is present only in human being or animals. And cellulose is only present in the plants. So that is the difference between the cellulose and the glycosin. Okay. Occurs in animals mainly called also called as animal starch. Yes, this is also called animal starch. Similar to amylopectin, that means water में soluble नहीं होते हैं. They are present in liver, muscles, brain, and also found in yeast and fungi. Yes. So with respect to biostrength, these points are important, but chemistry, it won't be asked. It has not been asked ever. अभी तक तो नहीं पूछा गया कभी भी. So okay, let's see. This is glycosin. Okay. Here C1 C6 linkage will be there as a starch. I have told you. So as a amylopectin I told you. So obviously it is quite clear that in the straight chain it will be C1 C4 linkage, but in the branch it will be C1 C6 linkage. So I hope it is clear. Yes. Now our next topic is protein, and that we will do in our next class. So I hope whatever I have explained that it is clear. Okay. So if there is any doubt, feel free to ask. And in next class we will start with the proteins, and we are around like to finish this chapter also. It was a small chapter, so the main part of biomolecules we have already covered. Okay, that was from the carbohydrate. So we'll do in next class. Till then, thank you class. Thank you so much.